Today we are starting a series of working through a prac paper. Now at AS Bio, obviously you're going to be doing the prac yourself, but that doesn't mean that you can't use the past papers for revision. The trick is to use the paper in conjunction with the instructions. So what we're going to do is we're going to work through this full paper. It's probably going to take us more than just today's lesson. So we'll do it in pieces, and then you'll see how it all works together and how you still get valuable information from working through like this. Also give you a good idea of how much of that paper you can do in your exam without even doing the prac. So you've got some good skills-based stuff that's being tested, not just your ability to do an experiment. So we are working in summer 2018, paper 31. And you always start at the very beginning. Before you proceed, read carefully through the whole of question 1 and question 2. Plan the use of the 2 hours to make sure that you finish all the work you would like to do. If you have enough time, think about how you can improve confidence in your results, for example, by obtaining and recording 1 or more additional measurements. You will gain marks for recording your results according to the instructions. So in other words, follow the instructions. But it's quite an important thing to think about that you read the whole of question 1 and question 2. Even though you're sitting at a microscope or you're sitting at a wet prac station, you need to be aware of the other question as well. Because remember, if you finish early on your station, you can work anywhere in the paper. So bear that in mind as you go through. You're not sort of sitting only with 1 hour for question 1, only with 1 hour for question 2. You can mix between the two. So what is our question? Milk contains proteins, which are used to make cheese. Okay, so we now thinking in terms of proteins. During cheese making, bacteria are added to the milk. The bacteria change the pH of the milk to acidic, causing the proteins to coagulate or clot, forming curds. The curds are then used to make the cheese. So we've got a few things here that we've come across before, but nothing that we've studied directly. So we've got proteins, we've got changing pH, we've got coagulation. So there's things that are very sort of familiar, but not quite in specifics. So we're going to need to keep our brains turned on as we go through here. So milk plus bacteria, the pH gets more acidic and the proteins in the milk coagulate. Estimating the protein concentration in milk is important when making cheese. And now we get to the crux of the matter. Estimating protein concentration. Okay, so we've come across things like estimating reducing sugar concentration. So this is something that we're familiar with. Okay, but estimating protein concentration is a new one. So they tell us though exactly what we're going to have to do. You will need to make a simple proportional dilutions of the proteins in the milk. Carry out the burette test on each concentration to provide a measure of the concentration of proteins present in the milk. Carry out the burette test on milk with an unknown concentration of protein, U. Estimate the concentration of milk protein in U. So we're getting our standard concentrations that we know, and then we're going to compare this unknown and say, where does he fall in relationship to all of those other ones? This is a process that we know about. This is something we're more comfortable with. So the question is now, how are we going to do it with milk? How are we going to follow the instructions of what they tell us to do? So we're provided with some materials shown in table 1.1, and you always want to go through and make sure that you have got all this present on your bench and everything is in order. So we've got M, which is 1% milk. No hazard, you got 40 centimeters cubed. You've got U, which is an unknown concentration of milk. You've got K, which is 5% potassium hydroxide. And you've got C, which is 0.15% copper sulfate. And you've got W, which is distilled water. And you've got lots of it. Okay, it's recommended that you wear suitable eye protection, always thinking about risk. And if K comes into contact with your skin, wash it off immediately under cold water. And something you might notice here, they told us we're going to be doing a burette test, but there's no burette solution. But remember, these two together, potassium hydroxide and copper sulfate solution, you can use those to do a burette test. You don't have to have a pre-mixed reagent for a burette. Right, so we've got our known milk, our unknown milk, our burette test, and we've got water. So we're pretty straightforward and set out with what we're going to be working with here. Then we get into the questions. You are required to make a simple proportional dilutions of the milk, of the proteins in the milk M 1%. Reduce the concentration by 0.2% between each successive dilution. You will also make a 0.1% concentration, and you'll need to prepare 10 centimeters cubed of each concentration. So the table shows how you're going to do 1% and 0.1%. Decide which other concentrations of milk to prepare using simple dilutions of M. Complete table 1.2 to show you how you're going to prepare the other concentrations. So, simple dilution. This is the one where we're going to have even gaps between each of our concentrations. It's not the one where we go by 10%, 10%, and then it gets smaller and then it gets lots more decimal points. Okay, this is the one where if this is a 2, then this is a 2. How much is it? It's a 0.2. So we're going to have a 0.2 between each of our concentrations. And we're going down to a 0.1. So if we think about it, we've got 10, we've got 0.8, 0.6, 0.4, 0.2, and then we're going to end up with a 0.1 that they told us we're going to do. So we're not too worried about the fact that this is 0.2, 0.2, 0.2, and then there's only a 0.1, because they've told us that we have to make that 0.1%. So it can be a really good idea to have these little notes on the side to keep track of where you're going. Don't go straight into your table, because there is something else to bear in mind. When you're sitting at your prac, obviously you've got all your equipment in front of you as well. And the number of test tubes that you have can be a good indication of how many dilutions you need to do. So if we go now and we have a look at the instructions. So for every prac paper, there is a memo, and there is also a set of confidential instructions. So these ones are released when the papers are released, and they have the full instructions. So the first bit is always about your microscope. And then they give you a little note about what size a test tube is, because remember, they've got to make sure that everybody's consistent across everywhere. Then we have, for both questions, a ruler, and then what we have, clean and dry apparatus, and then solutions, how they're provided, and then fresh solutions, materials, rinsing water, whatever is needed. Okay, so they're very detailed in exactly what must be provided. And here is the magic. For question 1, each candidate will require, and there is the full list of everything that's going to be on your bench. So here at the top, here's our M and our U, okay, that we talked about. Here is our K and our C for our burette, and there is our distilled water. So all of those are our solutions. Okay, so we've also see here that we've got another solution on our bench, which is hydrochloric acid. So we haven't come to that one yet. Then we get all of the equipment. So we have 2 10 centimeter cube syringes, 2 or 3 centimeter cube syringes, 1 centimeter cube syringe labeled C. And then we have test tubes. There are 7 test tubes. And a test tube rack, and a bung, and a spotting tile, and black card or paper, and a glass rod, a pipette with plastic or with teeth. So they were looking at a dropping pipette. Then we have our wash and waste beakers. We have paper towels, a glass marking pen, a stop clock, and suitable eye protection. So the piece of note that we want to look at now is this number of test tubes. Because if we go back here and we have a think, there's 1 concentration, 2 concentrations, 3, 4, 5, 6 concentrations, plus our unknown makes 7. And magically enough, there are 7 test tubes. So you can develop this habit of checking numbers even when you're working just with a past paper, because you can see what equipment do I have on my bench. So there is also nothing wrong with getting yourself another piece of paper and you've got your 7 test tubes. Okay, you can have 1, 2, 3, 4, 5, 6, 7 test tubes. So we know we're correct with our 6 concentrations, and we've got our 7 test tubes, 6 concentrations, and a U. So complete table 1.2 to show how you're going to prepare the other concentrations. The thing is that even though there's no lines there, we're going to have to add them in. So we add lines in with a pencil. They just don't want to give us any indication of how many concentrations we should be doing. But we now know we're adding another 4 concentrations in. So these are also done with a ruler, hey, when you do them on paper. Then we shall head back to our pen, dark blue or black. So we know the percentage concentration of milk that we're going to do is 1, 0.8, 0.6, 0.4, 0.2, and then there's the 0.1. So how are we going to do these? Well, what's quite nice is there's a pattern always for it, simple. 10 and 0 makes 1. 1 and 9 makes 0.1. So 2, 4, 6, 8. See how they've gone to 1 decimal place? So do we. We always match. So 8 plus 2 makes 10. 6 plus 4 makes 10. 4 plus 6, 2 plus 8. Because remember, we wanted to make 10 centimeters cubed of each concentration. If you are not happy with finding patterns, or if you want to calculate one, you can always say the 0.8 that I want times the 10 centimeters cubed that I want. So this is my concentration times my volume over my starting concentration, which was 1. And that's going to give me 8 that I need. So that's my volume of M that I need. So there's my 8. Then I add my 2 to get my 10. So if you do this one here, you say I want a 0.6. So you would have, I want 0.6% concentration. I'm working with a total of 10 centimeters cubed. I'm dividing by an initial concentration of 1. And that's going to give me 6 centimeters cubed of M that I need. So you can work out each one if you prefer to work with a formula, or you can look for the pattern. So you can do a couple and then do the rest by pattern if you like to mix the 2 approaches. Are we happy with this simple dilution? They tell us to read step 1 to step 9 before we proceed. And it's quite important that even though you've read through the entire paper to start with, you read these 9 steps again before moving on, because you want them fresh in your brain of what's going on. So, prepare all concentrations of milk as shown in table 1.2 in the test tubes provided. So we've got our 6 concentrations. Okay, remember we had 2 10 ml and 2 3 ml and a 1 by C. So you're starting to think now in your head, which syringes would I use for this? Okay, put the bung into one of the test tubes and invert it to mix well. Repeat with each of the test tubes, remembering to clean that bung in between. Label the spotting tile with the concentrations of milk prepared in step 1. So we would have our spotting tile, and we would have 1, 0.8, 0.6, 0.4, 0.2, 0.1. Use a pipette, so we're dropping, to put 2 drops of 1% milk into the labeled cavity on the tile. Any milk remaining in the pipette should be back in the test tube, so as little of the milk as possible is removed from the test tube. You're going to need it for step 13. So they're telling you here, don't waste. Repeat step 4 with each of the concentrations of milk. So now you would have all your concentrations of milk ready to go. Put 1 or 2 drops of K. We don't like instructions like that, okay, because that is inaccurate. It's not precise. Into each of the concentrations of milk on the tile and mix. So then put 5 centimeters cubed, 0.5 centimeters cubed of C into each mixture on the tile using the syringe labeled C. So here again, we're putting more liquid. So this is something why we want to read these steps 1 to 9, because we've got to think through in our head, I mean, how much liquid are we putting into each of these drops? Okay, leave for 2 minutes for the color to change. We're going to have to time that with a stopwatch. And then we're going to compare the color with the standard colors in figure 1.1, recording the color of the mixture in A part 2. A part 2 is simple and straightforward. Record your results in an appropriate table for the known concentrations of milk using only the standard colors in figure 1.1. So we've got to use these colors only when we're describing our results. So let's head back here and let's think again. So we had 2 by 10 ml, 2 by 3 ml, 1 by copper sulfate, and how many droppers did we have? We had only 1 dropper. So you see now that we're going to have to do some cleaning. So 2 by 10 ml, if we look at our volumes of M and W that we're using for our dilution, then these are our 2 by 10 mils. So we're going to have 1 10 ml for M and 1 10 ml for W. Okay, so now we've got our 2 by 3 mils and we've got 1 by dropper and I've got a 1 by C. So use a pipette to put 2 drops. So there's our dropper. Okay, we're going to have to rinse that dropper out each time because we don't want to cross-contaminate our concentrations. Then put 1 or 2 drops of K into each of the concentrations of milk on the tile and mix. Okay, but now we'd rather not have the dropper going into K and into our milk. So let's keep this one in the back of our heads. See, there's our C syringe. And then we're leaving for 2 minutes. So at this stage, we've got 2 3 ml syringes that we haven't used yet. So let's have a look quickly, because remember we came across that hydrochloric acid when we were looking at what's on our bench. So in theory, we would want 1 dropper just in there. Does that mean 1 syringe for hydrochloric acid? So potentially we have a syringe available to use just for our K. So let's just have a flick through and see what's going on. And there's a problem. Sample U. We need a syringe for U. Okay, I'm going speed reading further. And there we go. Hydrochloric acid. Putting 10 centimeters cubed of U. Putting 2 centimeters cubed of A. There's our other syringe. But note there we've got a 10 centimeter cubed of U. So if we go back up here and we think about how we want to minimize cross-contamination, we would then say for K, we can use a 3 ml syringe here. The other one would be for our hydrochloric acid, because we would rather use a milk syringe for our U. Plus we know we're heading to a point where we want to use 10 centimeters cubed of U, then we need quite a lot of it available. So when it comes to testing U, we're rather going to use our M syringe. Okay, yes, we're going to rinse it out and everything, so we have that same no cross-contamination, but U is simply another concentration of M. So we can use that same syringe. So now we are happy. Okay, we know exactly what we're going to do for this experiment, and we go ahead and do it. So we make all our concentrations of milk. We invert and mix well for each of them. We label our spotting tile. We put 2 drops of each concentration into its relevant spot on the tile. Remember, a spotting tile, the really nice ones, have actually depressions on them. So it's not a flat tile. Those little depressions, it's almost like a miniature bowl. And so the liquid stays inside that depression quite nicely. Okay, so we've put our 2 drops in each one. We've saved our milk. We've been sensible about how much we use, and we haven't wasted any. We've rinsed our dropper in between. And there we put, and we're going to decide, am I going to put 1 or am I going to put 2? Because you want to make sure you have the same in each one. And we're going to mix. We've got a glass rod, so we can mix nicely. Okay, we then put our 0.5 centimeters cubed of C into each mixture on the tile using its special syringe, and we leave for 2 minutes.